Hello everyone, and welcome back to another remote sensing lecture video. And in this video, I want to continue our discussion on spectral response curves slash spectral profiles. Right, in this case, I'm using the term basically interchangeably, but if you remember in our previous video, right, we outlined that there is actually a pretty big difference. And it's dependent on whether or not you're measuring the continuing spectrum of wavelengths, or if you have these sort of discrete bands and pixel values coming out of an image. But anyway, let's go through some more examples of these curves. Again, sort of reiterate what they're showing, how we can interpret them. And then in particular, I want to compare how spectral response curves look when you're comparing different types of objects to how different spectral response curves will look if you have different samples in the same object. And sort of hopefully hammer home, again, this whole idea of a spectral response curve. So what I want to do is I want to look at this sort of monstrosity right here. And when you first look at this, it might be a little bit overwhelming, but all this is showing is this is just a spectral response curve of 6 different real-world objects. Now, this is taken from real-world data, so these are actual values. I didn't make these up. Right. And so what we have is we have water, asphalt, 2 different types of vegetation, wet sand, and concrete. And again, the idea here, I'm just trying to hammer home that different objects interact with electromagnetic radiation differently at different wavelengths. I know that was a lot of difference, but basically what I'm getting at here, and I'm hammering this point home, right, if we were to take 590, right, these all have different values from each other. And if we were to take the lodgepole pine and go along the axis, right, so we're starting at 455 nanometers. Right, as we go along, you'll see that it goes up and down. Right, it's not a straight line. Now, water and asphalt aren't necessarily the best examples here because they're fairly flat, which means they don't interact that differently across the spectrum in terms of reflectance. Now, they may be absorbing, they may be transmitting. Right, we don't know the other 2 components, but in terms of reflectance, they behave pretty similarly across the spectrum. But again, just to reiterate here, I know I'm beating this to death, different objects react with different wavelengths of light differently. And that's why at a particular wavelength, we'll see different values for the different objects. And as you go along the x-axis for a given object, you'll see that the values change. That's all that's saying. So how can we start to sort of interpret these graphs? Well, if we look at, let's say, aspen, for example. Right, so down here, this is 455 nanometers. So this is, this region, generally speaking, right, this region is going to be blue. All right, so if we sort of do this, or you might think, okay, aspen is going to have a fairly low reflectance in the blue. So that means that most of the blue wavelengths are either being transmitted or absorbed. Again, we only have 1/3 of the puzzle. So then you might look and say, okay, well, in the red region, what's it doing? And that's going to be, from roughly, 600 nanometers to roughly 700 nanometers. Okay. So what's it doing in the red? Okay, so you look at aspen, it's also got, it's pretty low reflectance in the red. And again, I talked about this in a previous video, right? The reason that the vegetation, right, both aspen and lodgepole pine, the reason that their reflectance is relatively lower in the red compared to the green is because of photosynthesis, right? There's an actual molecule inside of the leaf of the plant that is actively absorbing, right, blue and red to use to catalyze photosynthesis. Right. So then as we move along, right, we start getting into the near infrared, right, and then we start getting into shortwave infrared, and you can see that we have this huge spike in vegetation, right? We have this huge spike in vegetation, right? Let's not use that. Let's use purple, right? So going from here to here, right? There's this huge spike in reflectance in vegetation. Right, and this huge difference right here, this is called the red edge. Now, this is unique, right, to vegetation, right? You can see that both vegetation types have this, right? The evergreen, which is the light green, this is the pine tree, as well as the deciduous right here. This is the aspen, right? They both have this huge spike that goes as you transition from red to transitioning to near infrared, right? And we're going to talk about things called vegetation indices and indices in general later. But one of the big key points of using these spectral response curves and spectral profiles, right, is we can start to see these sort of critical feature changes. And so we can do math. Again, we're going to talk about all of this later, but we can do math that compares, these wavelengths to these wavelengths. And we'll find that the difference here, right, is unique to vegetation. So we can use that difference as a way to discriminate or detect, right, vegetation from, say, sand and concrete, where the values don't change much over that range, or water even, right? So we can start to use these spectral profiles and spectral response curves to sort of figure out, okay, well, what are these objects doing over the entire spectrum of radiation that we're detecting? And are there any specific things that we can manipulate to help ourselves in the long term be able to highlight these things in an image? And we're going to spend an entire week talking about highlighting things in imagery using exactly these concepts. And that's why I wanted to show you, in particular, this graph here, because it really shows how these different things react differently and how we can use that information to make decisions. And we're going to go through another example specifically talking about how we would pick things in a later video. But for now, again, sort of we can look at each one of these objects as we go along the x-axis to see how they react at different wavelengths. And if we see any unique features, right, that separate things out, that can be extremely useful later on. So this is an example of looking at 6 different types of objects. Well, what happens if we were to take samples, right, spectral response curves of different individuals of the same type of object? Okay. So this right here, this is 5 different samples of spruce leaf. Okay. So we went out into the field, we got 5 different spruce leaves. So it's the same object, right? They're all spruce leaves. And we took their spectral response curve, right? So the x-axis here is wavelength, the y-axis again, percent reflectance. So what you might notice here is that they're all very similar in shape, right? Low reflectance, the red, this bump in green, low reflectance in blue, the bump in green, low reflectance again in red, this huge spike we just talked about, this red edge, right? And you'll see that these are all very similar, right? These are all very, very similar, right? Then we spike up. Actually, I probably shouldn't use purple there because we have a purple curve. Let's use black. Okay, right? These are all very similar right here. This is very similar. Right, we spike up, and then in here we have a lot more variability. We have a lot more variability up here. And so what this is kind of indicating here is that there's something that's occurring down here that is keeping things very tightly controlled, versus there's something up here that is going to be a lot more variable between the different samples. Right, and then we go on here, and there's a little bit of variability, but right in here there's a lot more variability. And now I'm not expecting you to immediately know the answer to this, so I'm just going to kind of walk through what's happening here. Right, these down here, right, this is predominantly controlled by photosynthesis, as I've mentioned multiple times with vegetation. Right, the red, green, blue, this sort of shape here, right, that is dominated by photosynthesis. And photosynthesis is something that is very tightly regulated in plants. And it makes sense, right? It's the way that they're able to produce their energy sources to survive. So it makes sense that these are going to have very tightly controlled reflectance profiles for a given species. Right? So we go up here. Now we're in the near infrared, right? So this is NIR, right? And so I'm just going to tell you, right? One of the things that drives the NIR reflectance in vegetation is water content. Right? Water content is one of the big driving forces in the NIR reflectance of a leaf. Versus here, right? This is driven by chlorophyll. Right? So this is photosynthesis and chlorophyll. In the visible, in the NIR, right? This is water content. And so it kind of makes sense, right? Photosynthesis and chlorophyll is something that's going to be very tightly controlled in individuals in the species. So you'd expect them to be similar. Versus water content, right? That can be very highly variable, right? How much did it rain? Is that particular tree nearer to a stable water source, right? So you can imagine that water content is going to change far more and be far more variable than the chlorophyll content for a given species. Okay, so hopefully that makes a little bit of sense. The other big thing that I want you to take away from this, and this is actually probably the more important thing, right, is just to quickly sort of, I know I've kind of scribbled all over these, but just compare the general similarities in shape between these 5 samples of the same object, right, in this case, a particular species of tree. Compare how similar they are to this. All right, I'm going to slide back and forth for a second, hopefully not make you nauseous. All right. Hopefully you were able to get the sense that, ooh, I accidentally panned way too far. Let's go back. All right. Hopefully you get the sense that these are, even though there is variability, these are still far more similar to each other than they are to anything else. Right. So actually what I want to do is I want to codify this. So let's just quickly wrap this video up by codifying this point, right? Individuals of the same object will have very similar responses. Right. And this is again, right, E dot G, right, different leaves of the same species. Right. Objects of a similar type will have moderately similar responses, right? And we saw this when we compared the aspen tree to the lodge pine, right? They were both vegetation, so they were similar types, but they weren't the same species. And so you saw that you had, moderately similar responses. This would be different, for example, right? For example, different species of trees. Right, and then this one is probably the most important thing and the most important assumption we're making, right? Objects of different types will have dissimilar responses. Right, and this would be, for example, right, water versus trees. Right, so hopefully these 3 points, right, individuals of the same object will have very similar responses, but not identical. Right, objects of a similar type will have moderately similar responses. Right, that was those different types of tree species. And then objects of different types will have dissimilar responses, right? The water versus the tree. So hopefully our example spectral profiles and spectral responses really hammered these 3 points home, because these 3 points, right, these 3 points really underline every single analytical effort, right? Analytical effort that we are going to make in the entire rest of the course. We're sort of trusting that these 3 things are true. That if it's if it's the same sort of object, it's going to be very similar. If it's the same type, like vegetation versus other type of vegetation, it's going to be very similar but not identical. And if they're different types, they're going to have completely different responses. So I know there was a lot. Hopefully this made sense. And if you have any questions, please reach out. Thank you.